Greetings everyone! Welcome to a new episode of Mr. Bioinformatics. Today we're going to talk about the most essential molecules in our bodies, what ancient Greeks called proteus, meaning primary, or the first importance. Today we're going to talk about proteins. My dear viewer, let me tell you that protein is not just an ordinary food. It's not about the chicken wings from your favorite fast food restaurant, or the whey shake you drink before hitting the gym. Proteins can actually evolve on their own, adapt and mutate without needing instructions from our DNAs. Our bodies contain around 20,000 to 25,000 unique proteins. These proteins are encoded by our genes and perform a range of functions, such as providing structure, like collagen in our skin, or catalyzing reactions, like enzymes in our cells. So what are proteins? What do proteins consist of? Each protein consists of one or more chains formed by linking small molecules called amino acids. Each amino acid contains carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur atoms. The general chemical structure of an amino acid includes an amino group, which is NH2, a carboxyl group, which is COOH, a hydrogen atom, which is H, and the variable side chain, which is R. What happens when this variable changes? The variable changes many times until it makes 20 different standard amino acids. 9 of them are essential amino acids that must be obtained from our diet because the body cannot produce them, like leucine, valine, and tryptophan, and the 11 non-essential amino acids that can be synthesized by the body, like glutamine, glycine, and serine. There are many structures of proteins. For example, the primary structure, which is the linear sequence of amino acids linked by peptide bonds. The secondary structure refers to local folding patterns like the alphahelix and the betasheet, stabilized by hydrogen bonds. And the tertiary structure is the overall 3D shape of a single polypeptide, stabilized by many interactions. And finally, the quaternary structure, which it involves many peptide chains, forming a functional complex, as in hemoglobin. Conformational changes are dynamic transitions in the protein's 3D shape. Someone might ask, does a protein go through these structures one by one, like phases? Actually, it's not a step-by-step process, where a protein is only in one structure at a time. Instead, as soon as the protein is made, it folds into all these structures simultaneously to become functional. But what drives these changes in protein structure? Well, environmental factors such as pH, temperature, and interactions with other molecules can influence protein's shape, allowing to adapt to different conditions. Proteins are dynamic, constantly shifting and adjusting their structure. Understanding how the protein is shaped is very important because its shape decides what it does. Proteins help with many tasks, like speeding up chemical reactions, making muscles move, or carrying oxygen in the blood. For example, when muscles contract, proteins like actin and myosin change their shape as they slide past each other. This movement makes the muscle fibers shorter, creating force. These tiny shape changes are what allow your body to move. Scientists have spent years studying protein shapes because seeing their 3D structure helps them understand how protein work, create new medicines, and learn more about diseases. But how did they actually discover these complex shapes? In 1957, John Kendrew, a British scientist at the University of Cambridge, he began his work to determine the structure of proteins. He chose to study a protein called myoglobin, which is found in muscles and helps stores oxygen. Why did John Kendrew choose myoglobin for his study? Myoglobin was smaller and simpler than other proteins, making it easier to analyze. The more complex a protein, the more complicated its diffraction pattern became. What exactly do we mean by saying diffraction pattern? Kendrew used X-ray crystallography technique. It is a method used to study the internal structure of crystals by shining X-rays into them. But why use X-rays? When X-rays pass through a crystal. They scatter in different directions. By capturing this scattered light, scientists can produce a diffraction pattern that reveals the arrangement of atoms within the crystal. The clearer the crystal, the more precise the diffraction pattern, and therefore the clearer the structure. But before Kendrew used this technique, he needed high-quality crystals of myoglobin. At first, he tried extracting myoglobin from muscles of horses and pigs. Unfortunately, these sources did not produce the clear clusters he required. Instead, the clusters were either too small or lacked the perfect structure needed for high-resolution analysis. Kendrew realized that marine mammals, like whales, had a higher concentration of myoglobin in their muscles because these animals dive underwater and they need to store more oxygen. Whale muscles are incredibly rich in myoglobin, and the crystals from this source were large and clear enough to produce detailed X-ray diffraction patterns. But growing protein crystals remained a major challenge. Why? Proteins are delicate molecules, and forming large stable crystals is a complex, time-consuming process. To extract the atomic positions from these patterns, scientists needed to calculate the phases of the X-ray, which is a highly complex mathematical task. So Kendrew relied on a method called phase determination, which involved comparing the diffraction patterns of crystals that had been modified in different ways, such as by replacing certain atoms with heavier ones. By making these changes and comparing the diffraction patterns, Kendrew and his team could figure out the phases, and lastly, the 3D structure of the protein. But here one problem remained. This process was too slow. To speed up the process, Kendrew turned into new developed electronic computers, the EDSAC, or Electronic Delay Storage Automatic Calculator. Was one of the first computers that could store and process large amounts of data. The EDSAC was built in 1949 and was one of the first stored program computers. Let me tell you, my dear, that it operated at a speed of about 615 instructions per second, or IPS, which was incredibly fast for its time. A modern smart processor like the Apple A17 Pro or Snapdragon 8 Gen 3 can execute trillions of instructions per second, or TIPS. Despite some initial skepticism, Kendrew's team began using computers regularly, and by 1962, Kendrew and his team had measured and analyzed over 25,000 X-ray reflections from the myoglobin crystals, a task that would have been nearly impossible without computers. Understanding myoglobin's structure revealed how its 3D shape enabled oxygen storage and released into muscles. This discovery led John Kendrew, along with Max Perutz, who worked on the structure of hemoglobin, to win the Nobel Prize in Chemistry for their groundbreaking work on protein structures. As scientists uncover more protein structures, they realize the need to organize and share this information to advance molecular biology. This is where a protein database or protein banks became essential. My dear viewer, in protein banks, they don't treat proteins like currency. Gene banks are digital repositories for 3D protein structure. First established in 1971 with the Protein Database Bank, or PDB, which has since become one of the largest resources for protein data. In 1980s, Swiss scientist Kurt Vortersch used nuclear magnetic resonance, or NMR spectroscopy, to study protein structures in solution. This method avoids the time-consuming and difficult process of crystallizing proteins. But how does NMR work? And why it is important? NMR detects the magnetic properties of a certain atomic nuclei, like hydrogen, which are abundant in proteins. By subjecting a protein sample to a strong magnetic field and analyzing the way the nuclei interact with the field, scientists can determine the 3D structure of proteins without the need of crystallization. This allowed us to study the proteins in more biologically relevant environment, as proteins in living organisms organisms rarely exist in crystalline form. These efforts helped lead to the development of cryo-electron microscopy, or cryo-EM, a method where proteins are quickly frozen in a thin layer of ice. This keeps the proteins close to their natural state, allowing scientists to study their structure without needing staining or crystallization. It has become a key tool in the structural biology, allowing researchers to visualize biological molecules in their near-native states by capturing images from different orientations and using computational algorithms to reconstruct the 3D models, including those of large and complex proteins that may be possible to crystallize, not like before where we obliged to choose proteins like myoglobin. By using computational techniques, scientists could create visual models to predict how proteins would fold and their eventually 3D structures. For example, the computational models of the p53 protein, a tumor suppressor, where simulations helped researchers hypothesize how it interacted with DNA. Additionally, in Alzheimer's research, simulations helped identify potential drug molecules that could interact with the beta-amyloid, a protein linked to the plaque formation in the brain. You can watch our sleep episodes to understand amyloid and what it does to the body. With all the insights gained from computational models, the next crucial question arises: how do we rapidly test thousands of potential drug compounds to find those that interact with these complex proteins? In 1951, Dr. Gula, a Hungarian scientist, he introduced the microtiter plate. Imagine a grid of tiny wells, each one a miniature laboratory, capable of holding many amounts of liquids. Before this invention, experiments were performed one by one in test tubes, consuming large volume of samples. But in 1980s and 1990s, pharmaceutical companies like Pfizer, Merck, GlaxoSmithKline embraced automation to quickly identify treatments for evolving diseases. Instead of testing one substance at a time, scientists could now process thousands of compounds in single run. This breakthrough method became known as high-throughput screening, or HTS. What is HTS? HTS is a high-speed method used in drug discovery and genomics to test many libraries of chemical compounds or biological entities for specific activities. It combines robotics, miniaturized biochemical assays, and AI to quickly analyze results. But how does it work? Firstly, a biological target such as an enzyme or infected cells is selected. Next, miniaturized assays are designed to test how compounds interact with this target, such as adding molecules to infected cells to see which one kills the virus without harming the cells. Robots then dispense precise amounts of chemical compounds into hundreds or thousands of wells for parallel testing. Afterward, sophisticated detectors measure the response, and AI algorithms go through the data to identify the hits. Hits are compounds with promising activities. For example, breakthroughs such as development of imatinib or Gleevec for leukemia and various antiviral drugs for HIV and hepatitis C were accelerated by HTS. HTS does not determine protein structures, but it's helpful when combined with methods like X-ray crystallography, NMR, or cryo-EM. HTS helps find which compound interact with biological targets, giving a starting point for further analysis. Once promising compounds are identified, techniques like X-ray crystallography and others reveal how they fit into the protein's structure, aiding the drug design. In 2020, DeepMind AlphaFold, an AI, revolutionized protein structure prediction by accurately forecasting the 3D shapes from amino acid sequences. This breakthrough has significantly impacted drug discovery and disease research. If you're wondering how incredible this achievement is, then let me tell you that in 2024, Demis Hassabis and John Jumper, they received the Nobel Prize in Chemistry for their contributions to protein structure prediction and design. If you want a full episode about AlphaFold to know how it's amazing and different from all other old methods, write AlphaFold in the comments below. Don't forget to like this video and subscribe to our channel. And here we've reached the end of our episode. Thanks for watching. See you next time.